All right, guys, the purpose of this video is to talk about the electrical conduction system in the heart, how we generate heart rhythm, how we regulate heart rate, and we're going to try to tie it all together. So this diagram is showing you this electrical conduction system. It's basically a network of cells that are not truly heart muscle cells, and they're not truly neurons. They're sort of a weird hybrid of in between. But the important thing is that they're able to generate and spread electrical signals or action potentials, just like nerves do, but they also have some heart muscle cell-like properties, right? All of this starts in a bundle of cells called the SA node or sinoatrial node that you find in the wall of the right atrium, right? We call the SA node the pacemaker of the heart because it sets the rhythm, right? I'll explain exactly how that works in a little bit, but for right now, just know that every time your heart contracts, it's because your SA node sent a signal, an action potential, that spreads as a wave of electricity across the heart, right? So when the SA node fires, we expect that signal to spread throughout the atria, right? And both atria will contract, right? This would be the right atrium. This would be the left atrium. So the atria contract because that's the first part of our heart signal. And then the signal also spreads to this thing called the atrioventricular or AV node. And the AV node plays a really important role. It pauses the electrical signal for just a tenth of a second. And the reason for that is the atrium and the ventricle, they have to take turns. So when the atria are contracting and pushing blood, the ventricle still needs to be relaxed, right? Because the whole point of the atrium is to pump its blood into the ventricle and fill it up, right? So we give that pause, we let the atria do their thing, they contract, they push their blood into the ventricle, and then a tenth of a second later, when the ventricle is nice and full, then the AV node will release its signal right down this bundle of His, it's sometimes called, or the AV bundle. And the AV bundle very quickly splits into two separate bundle branches, right, that fork to the left and right ventricles. So right bundle and left bundle are just left and right ventricles. And then from there, they branch further into this network of smaller structures that we call Purkinje fibers. And that whole network is contracting the ventricles, right? So all of this plays out. We see it on an ECG reading, and we'll notice that in a little bit in more detail. For right now, let's take a look at the SA node and exactly how it's generating that pacemaker potential. All right. This idea of a pacemaker, it really just means it's a rhythm generator. So no different than like a drummer in a band that's keeping rhythm. You have to have even spacing between heartbeats, right? And so when we talk about heart rate, like imagine I say that someone's heart rate is 60 beats per minute. We assume that those beats are evenly spaced. And if it was 60 beats a minute, you would expect to see one heartbeat exactly every second, right? We wouldn't want the heart to flutter really fast for a little bit and then slow down and be all erratic, right? We want consistency. That's what the pacemaker is giving you. It's effectively a self-timer. And so even if you disconnect the heart from the nervous system, like during a transplant, the SA node will still generate rhythm all by itself. That's why we say it's autonomous. It's happening entirely within the heart. You don't need the brain or spinal cord or anything else to make it happen. Right. Now, the mechanism, when you look at the membrane potential, right, same membrane potential that you saw in the nervous system, every action potential, right, every one of these spikes from the SA node is one contraction, one heartbeat, right? So every time you get a spike, that is a signal coming out of the SA node to start the cardiac cycle, make the heart contract, right? That's what I was showing on the previous image of the signal spreading from the SA node to the atrium and to the AV node. That is this. Now, the catch is, in a neuron, you typically have to use neurotransmitters to hit threshold to get an action potential. In the heart, we have this, when they say slow influx of sodium, this is basically channels that are leaky, right? Leaky meaning, I think of it kind of like an hourglass. When you turn an hourglass upside down, the sand leaks through and you use it like a timer. These leaky channels are leaking sodium, right? And the positively charged sodium is making you go up toward threshold. And when you hit threshold, boom, action potential, right? But you immediately repolarize back down and start the leaking process all over again. And so the fact that the sodium channels give you a steady, slow leak, that is what gives the SA node rhythm. Right now, the catch to this is you can change the speed a little bit, right? So you can adjust how fast you hit threshold. And for example, if I added epinephrine, epinephrine sympathetic nervous system increases heart rate, right? Its effect would be to open more of these sodium channels and make you hit threshold faster, so the next heartbeat comes a little bit sooner, heart rate's sped up. If I released acetylcholine, right, which is a parasympathetic neurotransmitter, that decreases heart rate, which means it would have the effect of making it take longer to hit threshold so that you have more time between heartbeats, slows your heart rate down, right? So the SA node is controlling the rhythm aspect of it, and it has a default speed. And then you can use your nervous system and your endocrine system, right, to release neurotransmitters and hormones that will either speed up or slow down the rate a little bit depending on what's happening. Right. Okay, so once our SA node sends its signal, we need that signal to spread throughout the heart. And cardiac myocytes, remember myocyte really just means muscle cell. So this is just a heart muscle cell. They contract in a very similar way to skeletal muscle, right? Same sarcomeres, same actin, same myosin, troponin, tropomyosin, all that stuff is the same. And so I'm not really going to go into it because you should have seen it in skeletal muscle. But the idea here is that we need all of these heart muscle cells to contract together. It's not like skeletal muscles where, depending on how heavy the thing you're lifting is, you activate different motor units. The heart is like a single unit, all or nothing. And so what we use is these structures called gap junctions. And gap junctions are effectively little channels between cells, so that if you activate one myocyte, one muscle cell in the ventricle, it's going to start spreading to all the other surrounding ones, because those ions that depolarize the cell can move through the gap junction, right? In addition to that, you have these intercalated discs that are made of desmosomes. These are really, really strong intracellular junctions that are just structural support. They're making sure that when the force of the heart is contracting, that you don't rip those heart cells apart from each other. Okay, so at this point, we've got our pacemaker potential hitting threshold in the SA node. It's starting to spread through myocytes. It's spreading towards the AV node, right through all these gap junctions, right? And eventually, we're going to get this wave of contraction across the entire heart, synchronously, all at the same time, right? So our heart cells are working in coordination with each other, right? Within an individual myocyte, the similarity here, remember in skeletal muscle, an action potential in skeletal muscle led to calcium release, calcium bound to troponin, that let actin and myosin bind to each other. All of that's the same here, and so I'm not going to rehash it. The difference is in skeletal muscle, the action potential is really fast, right? And that gives you a fast little twitch where tension shoots up and comes right back down. In a cardiac muscle, it's different. We need the cardiac muscle contraction to last longer. So instead of the sharp spike for a skeletal muscle, we want a long, relatively slow contraction. I know 300 milliseconds isn't that long, but it's an awful lot longer than 30 or 40 or 50 milliseconds, right? So the way that this happens is the calcium channels in heart muscle are different, right? The calcium channels are intentionally slow so that once you spike up, right, you hit threshold, it's the same voltage-gated sodium channels opening that you have in neurons or in skeletal muscle cells. And that sodium gets you the initial spike, the depolarization, but then you get this nice, long, steady plateau where calcium is flowing in, and that is what is causing the contraction, right? Calcium entering the myocyte stimulates it to contract, and eventually, once voltage-gated calcium channels open, which are incredibly slow, the potassium opening and the calcium channels closing give you repolarization, right? We come back down to normal. And so the general process is very similar. Sodium depolarizes, potassium repolarizes. You've seen all that before. The only difference now is these slow calcium channels that make sure that we hold the contraction for a longer period of time to make sure the heart has enough time to actually push all the blood out. Okay, so now's the time to sort of put some of this stuff together. This line down here is showing you an ECG. You will also sometimes still see it called an EKG. The K is because the Germans developed the ECG, and cardio is spelled with a K in German. It's the same thing. So in an ECG, we're putting electrodes on a person's skin, and we're measuring the electrical signals coming off of the heart. And so it basically gives us an average of all the electricity in a heart over time. Right? So if we extended it over here, eventually you would see another P wave. Right? And so the distance from the start of one P wave to the start of another P wave, that represents one heartbeat, one contraction cycle. And specifically, the P wave is responsible for making the atria contract. Right? And so this P wave that they're showing highlighted in purple, once that happens, we get this wave of depolarization. When you see the purple on there, think depolarize, and depolarization means you're stimulating it, so the muscle is going to contract. Right? So when the atrium is purple, we're contracting. Notice if you look very, very closely, this little purple region right here in between P and Q, that is the pause, right? That is the AV node pausing the action potential. That's why the line is flat, giving the atrium time to finish contracting so we can fill up the ventricles, right? Once that's over and the AV node releases that signal to go down our bundle branches, right, down into the ventricle, then we get this QRS spike, right? So QRS, and remember, these letters don't really mean anything. Someone just decided to name them that. I'm honestly not even sure why. And so P, Q, R, and S are just showing different inflection points on the graph. But this QRS spike is showing you the beginning of contracting the ventricles, right? And so ventricles are contracting. We're pushing blood up and out the aorta and the pulmonary trunk. And when you finally get to the T wave represents repolarization. And think of repolarization as meaning relaxation, right? We're resetting the ventricles, relaxing the muscle. And when they're relaxing, that means they're refilling. Once they refill, we come right back around, we have another P wave, and we start the entire cycle all over again. Okay? Right? And names that you'll see put on top of this, this is all lined up, so it shows you in time. So this area being atrial systole, systole just means contraction, right? So this is atrial systole. This region is ventricular systole or contraction, right? And then this gets into ventricular diastole, right? Because this is when the ventricle is relaxing. And then once we get another P wave, we're going to start the whole thing all over again. Okay. All right. Well, hope this was helpful. Again, follow along in the playlist. The next video in line is going to put a lot of this stuff together and look at ECGs and contractions and how it changes pressure and blood flow within the heart.